Okay, good morning. My name's James Danger, and I'd like to spend some time with you on a video in my embryology section looking at the factors of the prozygote that affect first cleavage. It's based on an article produced in Journal of Assisted Reproduction and Genetics in March 2026 by some West Australian authors. And basically, the paper is looking at the implications of the pronuclear form inside the zygote, and how that relates to the first cleavage division, and whether in fact that has any impacts on the ability of the blastocyst to produce a live birth. So just a little background stuff. There are two articles I want to just briefly raise with you just to set the scene. The first one is one from 2001. In development, this is where it's the blastomeres arising for the first cleavage division in mice have a distinguishable phase in normal mass development. And the authors say, thus at the two-celled stage, it is already possible to predict which cell will contribute a greater proportion of its progeny to the abembryonic part of the blastocyst, that is, the region including the blastocyst cavity or the trophectoderm, and which to the embryonic part, the region containing the inner cell mass, and ultimately the fetus and the embryo proper. And there's a second article, which is a bit more complicated, published in 2017 in Scientific Reports, looking at the totipotency suggests between sister blastomeres, the two-cell stage mouse embryos is variable. And they say, following fertilization in mammals, it is generally accepted that the totipotent cells are exclusive to the zygote and to each of the two blastomeres originating from the first mitotic division. This model of totipotency was inferred from a minority of cases in which the blastomeres produced monozygote twins in mice. And what the authors did, they separated the blastomeres of 1,200 off two-cell embryos, and they identified two classes of monozygotic twins becoming apparent at the blastocyst stage. 27% of these cells formed a functional apoplast in both cells of the two-cell embryo, but 73% or three-quarters of these embryos did so only one member of the pair. So in a quarter of the two-cell embryos, both blastomeres were able to give rise to a fetus and a baby, but in three-quarters of the cases, only one of the two blastomeres, the two-cell embryo, were able to do that. And that this is discordant, as they say. And this is a partition that proved to be insensitive to oocyte quality, sperm entry point, culture environment, and a pattern of cleavage. So in intact two-cell embryos, the ability of sister blastomeres to generate the epiblast, which is the early fetus, was also skewed. Class discovery, clustering of individual blastomeres, and the blastomeres transcriptomes, that is the protein production, points to an innate origin of concordance and discordance rather than something being acquired during development. The data places constraints on the commonly accepted idea that totipotency is allocated equally between the two-cell stage blastomeres in mice. In other words, the two blastomeres of two-cell embryos are not the same. And just to finish off with this paper, because I know it can be complicated, and this is really just to sort of blow you out of the park. They injected one blastomere with a green dye and one with a pink dye and watched the embryos develop, and then counted the number of green and pink cells in the epiblast. And even though that each blastomere in a few cases contributed 50% of all the cells in the epiblast, the vast majority of each end of blastomeres only contributed all of the cells that is, all these individual blastomeres, they produced so even I'm getting tongue-tied with this, I apologize. That all these individual blastomeres contributed totally to the epiblast, and the other ones did not. So among the 36 labeled non-bisected embryos, 8 featured balanced proportion, that is, one blastomere supplied 50 to 56% of the total epiblast cells down here, right? And 14 featured markedly skewed proportion where one blastomere supplied more than 80% of the epiblast cells. So these these cells here, they contribute 100% of all the cells. So it's either the red or the green. 100% of all the cells in the blast only came from one blastomere. Now I have to warn you that parts of this talk are a bit complicated even for me. And if I get tongue-tied, it's because I'm still not entirely sure I have my head around all the aspects. As I've just demonstrated, it's not necessarily self-apparent what's happening. But the bottom line is, the message here is, and the basis for the paper is, that the two cells, each blastomere of a two-cell embryo is not necessarily identical, and that if the two-cell potency theory is not inherent but variable, what might give rise to the variability in the zygote, and can these be used to predict the outcome of embryo development? And therein lies the reason for this publication. So the purpose of this was to investigate whether alignment amongst the zygotic cleavage plane, CP, the pronuclear axis, and the second polar body axis were associated with a higher live birth rate in single transfer blastocysts. And alert, their conclusions were their findings provide preliminary evidence to support zygotic alignment among the cleavage plane, pronuclear axis, and polar body axis as a potential live birth marker for blastocyst selection. In other words, if you look at how the embryo alignment of polar body and pronuclei on the zygote and how the embryo cleaves relative to that plane, right from day 1 and day 2, you may start to be able to preferentially allocate embryos into may transfer to maybe not transfer. And right at the very end, I'm going to come back to this statement. Basically, what I'm trying to do is paint a picture of why I'm looking at this article. This article is a summary of many of the key zygotic events that occur out of sight. When all the key axes align like stars, such as the polar body axis and the pronuclear axis, and the cleavage plane is in alignment with these, then the embryo would more likely be euploid and more likely have the preferred cytoplasmic properties, then that they will more likely lead to pregnancy and live birth. So embryologists trying to work out which embryo to transfer don't necessarily have to wait until day 5. On day 1, they can already be allocating embryos to look at closely or unlikely to form a blastocyst. Okay. So the pronuclear phase is often ignored because of the small pronuclei are often dynamic and transient, and the time they break down is often viewed as a marker of viability. But this breakdown is often related to sperm DNA fragmentation and delays in processing the sperm DNA rather than aspects of aneuploidy and tatipotency. But more importantly, I think that more often the period before the zygote gene activation, which in humans is 4 to 8 cell stage, is considered as unimportant and dismissed. Most embryologists these days are only focused on euploid embryos, and everything else is of little consequence. But there have been several papers now that have returned to the pronuclear phase and found much of what happens on the first day will affect what happens later. That these effects can impact on the likelihood of pregnancy is very important and may be critical in identifying which, if any, embryos will implant and form a viable pregnancy based on what the pronuclei are doing. It could almost be a KPI, the number of pronuclear embryos where there is alignment. I apologize. And just the last of the preamble before I get to the data, I just need to basically talk about the pronuclei. And before I do that, I need to give honor to Lynn Scott, who's passed away several years ago. But Lynn Scott, a fantastic embryologist, did most of the development work in the 90s, looking at the pronuclei and the organization of the nuclei and the nucleolar bodies, and scoring these and showing that these did have impacts. So I think I'll discuss this paper at another time. This was looking at the difference between IVF and ICSI, which I think we'll have to keep coming back to because I don't think that they are identical. But even in this paper published in 2021, only 4 or 5 years ago, if you gave a embryo score of 1, which was this type here, 2 pronucleae closely aligned of equal size, the nucleolar bodies are 3 to 4 and closely aligned with the junction, then they are PN score 1, they're the preferred ones. And if you put those embryos back, they're more represented in the implanting embryos and all the other forms. In other words, these embryos the zygotes that form this pattern pronuclear development are more likely to implant and give you pregnancy than all the other ones. In other words, this formation is really critical. And just to ignore it, saying, oh, it's a big function of time, may be naive. And at this point in time, this has got to do with identification. I have to do a little plug for myself, so I do apologize. Self-promotion. This is my website, ferdaid.com, and I offer online EQA programs. But if we look at this one, the pronuclear assessment, which I do every year, HEB, every month I show you a picture of an embryo. These are often GIF files going through several different planes. I ask a series of questions: cytoplasmic inclusions, Scott's definition, halo effect, how many nuclei are there, the juxtaposition, how many are in each pronuclei, left or right, their synchrony. And then all these different options have different grading, like lots or little or no or whatever. But they all have values, which I add them all up, calculate to score out of 100, and I get a pronuclear score for this zygote, which is normally distributed, and that allows me to do quality assurance. So this is what, someone has completed this quiz, answered all these questions, and for each of these questions, you can see how accurate you are compared, or how you agree or disagree with everybody else. So this particular question here, juxtaposition of pronuclei, the staff members said strongly together, which of course they are, and 75% of people agree they were, but 25% of people did not agree. So this, if you can then basically calculate all these things, give you a score, and then the clinic can go back and look at the activity of each individual and the clinic compared to everybody else. You can see here, for instance, this one, that the clinic score of 11 staff members had a score of 85, I think, but the overall score of 34 participants was a 37, so they were half a standard deviation below the mean. And basically, all the clinic is actually scoring their pronuclei as less developed than the majority of other people. So in fact, if they were to publish a paper describing the pronuclear score, they would be actually scoring more embryos grades 2 or 3 than 1. So enough of self-promotion. Let's get back to the topic at hand. There was a poster in 2021 from some sort of Japanese authors who are starting to suggest that the 2PN axis is essential for determining the first cleavage plane, because it seems to be involved in the positioning of the mitotic spindle poles, centromeres. The direction of the first cleavage plane relative to the 2PN axis can be an important indicator in predicting embryo development and pregnancy. Another paper a couple of years later, also from some Japanese. The Japanese, I think, have spent a lot of time on day one identification. They spent more time than most other people looking at the implications of it. But this paper, it concluded the formation pattern of the first punter division relative to the pronuclear axis was a predictor of aneuploidy, with reduced rates of euploidy and high probability of aneuploidy was observed when the first punter division was perpendicular to the pronuclear axis. So here we're getting a sense that the relationship between the pronuclear axis and the first division, the cleavage axis, may be an indicator for the viability of the embryo, but here we're suggesting it's also looking at euploidy rates. So there was a poster produced in 2022, and that their paper came out 2 years later from Publikovas, and an excellent paper. So unlike in mice, they've argued that the human zygote tends to divide at right angles to the longitudinal pronuclear axis, irrespective of polar body position. And there was another paper back in 1999 suggesting that pronuclear orientation, polar body placement, and embryo quality after ICSI and IVF are different, and there is further evidence of polarity in human oocytes. And so once again, there's a fair amount of divergence of opinion between whether the IVF and generated embryos are the same or they're different. Okay. Finally, after this long preamble, I do apologize. You can probably stop and have a cup of coffee, then come back. Let's focus on what these authors have identified. This is an open access article, and they're looking at 3 axes. Might just pause just for a minute over. Hopefully that's cleared my throat. So the authors were interested in 3 axes. One was the polar body axis that runs through the second polar body, and this basically marks 2 parts of the oocyte. This is what they call the animal pole, and this is what they call the vegetal pole. The second axis is where the two pronuclei are abutting, and the axis through the two pronuclei is what they call the pronuclear axis. And the line between the two blastomeres, the tussle embryo, which is called the cleavage plane. And what they're looking at is the relationship between this, and whether that has any predictive power in a live birth. And they've produced a graphic to try and identify what they're doing. So they're comparing embryos where there's alignment between the zygotic cleavage plane and the pronuclear axis, yes and no, and alignment between the zygotic cleavage plane and the polar body axis, which is yes and no. And some of these are straightforward and some of these are a bit, I'm not quite sure I understand it, but in the simplest form, this is the polar body axis, this is the pronuclear axis, and this is the cleavage axis. So they all agree that's a yes, yes. But some of these ones here I just don't quite understand, and I think that it could have been a little bit clearer. But if I, for instance, go back to the no case, here is the polar body axis, here is the pronuclear axis, and here is the cleavage axis, and you see that they are the cleavage axis is not aligned. So they're really comparing this group, and these are the two alternatives. And their data is here. It's only 138 embryos, which I think is far too small for this sort of study. And hopefully they will come back with a second paper and maybe 500 or 600 embryos, which will add a bit more clarity. And I'm only going to focus on these two sets of data. This is where there's alignment between the cleavage plane, the pronuclear axis, cleavage plane, right, and in the live between the cleavage plane and the polar body axis, CPB, P+. In this case, is 58 out of the 138 embryos had complete alignment between the two, and of those 28 went on to deliver a live birth, almost 50%. And if you look at the group where there was an unalignment between the cleavage plane and the pronuclear axis and the polar body axis and cleavage plane, only 18 of the 138 embryos formed in this group. So this is actually quite a rare scenario, but only 2 of those 18 embryos formed a live birth. And all these different, you can see that the non-alignment between the cleavage axis and the polar body axis or the pronuclear axis gave you pregnancy rates which were still quite low, 19% and 16%. And they also represent around about half of all the embryos. So most embryos, a good third, will divide in complete coordinates with the polar body and the pronuclear axis. Only about 15%, less than 20%, they're contra-aligned. But most other cases, there's alignment between one or the other. So it's all a bit of there's a spectrum of responses. It isn't yes or no. It's just a huge variation between the two. And they went through and just produced some somewhat confusing data comparing IVF and ICSI and fresh blastocysts and frozen blastocysts. So this data set wasn't a pure data set. It was a combination one, which is both a strength and a weakness. But if you look at, and once again, it's not sort of separated in the 2 by 2, but if you look at alignment between the IVF and ICSI, basically you can see in their data they are very similar. So they've drawn the conclusion that the IVF and ICSI procedures produce the same sort of outcome in terms of live birth. So unlike some other authors, they can't see a difference in live birth between the orientation of the pronuclei and the live birth. But there was a difference between fresh and frozens, and they haven't really talked about that too much. But 64% to 42%. So basically, the frozen blastocysts behaved at a slightly lower rate. And I'm not sure whether these frozen blastocysts are because they were frozen for PGT or whether they're frozen because they were surplus for the fresh transfers. I think I need back to read that paper again. But the trends here is that IVF and ICSI behave the same way, and fresh and frozens behave differently. And I think that variability needs to be clarified in a future publication, which I hope they're already working on. But the bottom line still is that the alignment for the polar body and pronuclei is 64%, 1 in 17, 61 to 12%, really different. And likewise with the polar body, likewise with the frozen blastocysts, and likewise with the frozen blastocysts with the polar body. So in their sort of data, the polar body and the pronuclei both appear to be independent predictors of the relationship between live birth. Okay, so what we're talking about is the alignment between the second polar body and the alignment of the pronuclei and how the embryo cleaves. So theoretically, according to their data, this is the preferred zygote you'd be wanting looking at, the second polar body, alignment here, and the two pronuclei all in the same alignment. And what they're basically saying is their data supports the hypothesis that the alignment between the cleavage plane, pronuclear axis, and polar axis are associated with a high live birth. These findings demonstrate that correlation between the zygotic alignment and live birth is independent of the insemination method, thereby bridging an important gap in the current literature. This is to some extent consistent with an early study based on static morphology, which reported that the pronuclear orientation relative to the polar body position was similar in human zygotes derived from both IVF and ICSI. But that's only really looking at the orientation between pronuclear orientation and the polar body. So now we're getting into something which I think is more important. Accurate chromosomal segregation during the first zygotic cleavage is paramount, and segregation errors may lead to aneuploid daughter cells. Therefore, it's speculated that the alignment between the zygotic cleavage plane and the pronuclear axis could promote correct zygote cleavage. The zygotic polarity is well established in animal models, with the second polar marking the embryonic pole as opposed to the abembryonic pole, although it remains unclear in humans if this is absolutely true. It's hypothesized that the misalignment between the zygotic cleavage plane and polar body axis may lead to an unequal distribution of polarized cytoplasmic determinants between the daughter cells, potentially impairing subsequent embryonic development. And at this point, I now to bring in two other articles which I think relate to this, both published by a group whose main author is Magdalena Zanika Goetz. One in 2004, one in 2005, both working human and mouse embryos. They're both open access, and everyone should read these two, although they're very complicated because they contain a huge amount of technical data. But if you read the introductions and the discussions, I think it's relevant for embryologists. First is, where they also conclude that the first two blastomeres contribute unequally to the human embryo, supporting what they're seeing in the mouse. And that is, if you do lineage tracing of human embryos from the two-cell stage to the blastome stage, the majority of the epiblast, that is the part of the inner cell mass that forms the baby, is derived from only one of the two-cell blastomeres. Early asymmetric divisions are a bottleneck controlling the embryo's clonal composition, and that is the first dividing two-cell blastomere generates more asymmetric divisions at the HS stage, which leads to allocation of cells into trophectoderm or inner cell mass. So there's a lot to do here, which I think is almost another topic of how the embryo divides from the 1 cell to 2 cell to 4 cell to 8 cells, and in so doing start to force embryos to either the outside of the embryo, inside of the embryonic mass. The second paper I think is more important, although it's more problematic, and that is they looked at the protein symmetry inside the fertilized oocyte, and here they say proteomic asymmetries emerge at the zygote stage and are triggered by fertilization. Two-cell stage blastomeres cluster into alpha and beta states with unique profiles. These are just two unique subsets of proteins, which they call the alpha and beta. That doesn't mean anything. There's no primacy attached to that. But the beta blastomeres show greater development potential than the alpha blastomeres, and early asymmetries are conserved in human embryos. So what they see in mouse, they also see in humans. Basically, what this means is, they basically say if you take the two-cell embryo and allow those embryos to develop, you mark them in some way, is that one of the two blastomeres will end up predominating the cells in the inner cell mass and the epiblast and give rise to most of the fetus, while the other blastomere will, even though some of it may be in the epiblast, the majority will be in the trophectoderm and give rise to placenta. But more interesting for me, this is a paper really caught my eye, was if you look at the protein symmetry, you do complex protein analysis, which is, it's a little bit beyond me. but you can get the sense of what they're talking about. You don't actually need to be a biochemist or a geneticist to really follow their argument. Is that this graphic, I think, actually demonstrates it all, that there is increased asymmetry. That if you look at the fertilized egg relative to where the sperm is fertilized, there is an asymmetry across the zygote in terms of proteins being produced. So all these proteins are produced as a consequence of fertilization, as a result of the phospholipase induction of calcium oscillations, setting in train a whole process of things that's happening inside the zygote that you cannot see. But the consequences is that how the zygote then cleaves may then impact on the proportion of the different sorts of proteins in the two blastomeres, where they're called alpha and beta. And this diagram implies that one blastomere contains more of the red or beta protein profiles, and the second blastomere contains more of the alpha type protein. And consequently, when the embryo divides, some blastomeres are highly influenced by the alpha protein subset and the other ones by the beta subset. And when you go back and look at the distribution of it, that the blastomere with a large amount of the beta protein, that is this embryo here, gives rise to a larger blastocyst and a larger epiblast, this group here, which gives rise to the fetus. But where you have a different sort of asymmetry, where you have far more of the alpha type protein mixed than the beta type, you still get a blastocyst, but you get very little epiblast. And I have to say, if I look at that, so you can see how the variability in tadipotency may arise from the degree that the two protein profiles that develop in the zygote after fertilization are contained or dispersed within the cytoplasm. And this has implications is that the orientation of the pronuclei in the protein matrix may be linked, or how the zygote divides will determine the relative separation of protein gradients. So if the embryo divides along the polar body axis, the pronuclei are aligned this way, you get a very clean separation between the two blastomeres. And where the alignment is at odds, angle, or perpendicular to this axis, you get a confused set of signals inside the thing. And just as if I look at this embryo, compared to this one would be like a 4AA type embryo, but this one would be like a 4BA or a 4CA. So if you look at the size of the inner cell mass, you can see that this author is implying, at least in the mouse and maybe in the human as well, that the disproportion of alpha and beta type protein matrices will determine how many cells are allocated to the inner cell mass. And there are other publications come out recently where looking at predictors of live birth. The embryos that have the highest live birth are the ones that have the biggest inner cell mass. So 4A embryos will always do better than 4B embryos. Okay. Let's just look at a couple of the papers because I'm running out of time and running out of water. I need a coffee. Here is a paper published in 2019, Scientific Reports, saying the fertilization cleavage axis differs in primates between IVF and ICSI. And I think this is just an ongoing debate, which I think will take a bit more time to resolve. Multiple videos of ICSI show atypical sperm head displacement between the oocyte cortex and eccentric paratangential pronuclear alignment compared to the IVF zygotes. Neither fertilization generates incorporation cones. At first interphase, opposed pronuclear align obliquely to the animal vegetable pole after ICSI, with asymmetric furrows assembling from the male pronucleus. And so there is some, I think this, even though the main paper didn't show any difference between IVF and ICSI, I still think there are lots of papers suggesting there is something there between how oocytes are fertilized with IVF and ICSI, which needs our attention. And there is another one from Hubakova, which I think is a really great paper, also very complicated to read, but they start to suggest a relationship between cleavage and the nuclear status of the human embryo. In order to delineate the spatiotemporal organization of the first mitotic division, typical of normal human embryo development, they systematically analyzed a unique time-lapse data set of more than 300 IVF embryos that developed into healthy newborns, similar to what the original authors did of this. The zygotic division patterns of these best quality embryos were compared to their siblings that failed to implant or arrest during the cleavage stage. And they showed that division at the right angle to the juxtaposed pronuclei is preferential and supports faithful zygotic division. Alternative configurations at the first mitosis are associated with reduced clustering of nucleoli and multinucleation at the two-cell stage, which are more common in women of advanced age. Collectively, these data imply that the orientation of the first division predisposes human embryos to genetic instability or stability and may contribute to aneuploidy and age-related infertility. So you can see that suddenly the orientation of the pronucleus takes on a whole new meaning in terms of the euploidy-aneuploidy mosaic argument, which is dominating IVF. And I wrote to Dr. Hubakova, and she sent back this text, but basically we're trying to get head around what we're talking about. And I have to say, sometimes I understand completely, sometimes I still get confused. So if you're confused, I understand. But basically the way to think about it is this. If you look at the alignment with the two pronuclei, forget the polar bodies at the moment. What happens is when you look at the pronuclei with the nucleolar bodies, the alignment, the centriomes, which are originated from the male, appear at the junction of these two pronuclei. So they appear here and here. And the centriomes then act to the chromosomes, the nucleolar bodies then break down into the individual chromosomes when there's pronuclear breakdown, and they form a mitotic spindle, and the chromosomes are separated or segregated along this spindle axis, which is perpendicular to the pronuclear axis. So we're now talking about a geneticist point of view rather than embryologist point of view. So there's a pronuclear axis, and there is a spindle axis, which is at right angles to it. But when the embryos divide, you get a two-cell embryos where the cleavage furrow is in line with the pronuclear furrow axis. So in our study, the pronuclear axis is defined as a line connecting the two abutting pronuclei shortly before nuclear envelope breakdown. If chromosome segregation occurs perpendicular to the pronuclear axis, our perpendicular axis, this one here, and the cleavage furrow forms perpendicular to the spindle axis, then the cleavage furrow will form parallel to the pronuclear axis. This is why in figure 1c may appear counterintuitive if the term cleavage axis is interpreted based on the position of the cleavage furrow rather than the direction of the chromosome segregation. So if you're having trouble recognizing that we have to think from an embryologist's point of view, this is the angle here, and the embryo will split into two halves. But from a geneticist's point of view, it's to do with the spindle angle, which is perpendicular to the pronucleus. So what you really want is to have a pronuclear angle where the spindle axis is perpendicular to it, and a perpendicular axis will give rise to a parallel cleavage furrow. I apologize for spinning. I actually spent about a week trying to get my head around this. So I'm still not quite sure. But basically, if you take that image and run the same line, so you have a pronuclear axis running down here, the centrosome, the centromeres sit on either side of it, and pulling the chromosomes in, so the cleavage furrow is this way. So what you get is an embryo with the pronuclear axis, and the cleavage axis is the same. Right, but the spindle axis is this way. In this case, you have a cleavage where so even though this means that the cytoplasm here was splitting this way rather than this way, which means there was problems where the centrosomes were collecting. And so if you go then to the Hubakova data, in their summary, they looked at the arrangement with the polar bodies, and they could show that whether it was in line or orthogonal to the polar bodies, they had the same distribution of embryos and roughly the same number of arrested to live births. So they couldn't find an arrangement with the pronuclei. But if you look at the polar bodies, this is the distribution of the polar body in the cleavage plane, and this is what they call parallel, and this is what they call perpendicular. So this is perpendicular to the spindle axis, right? But you can see here that where the cleavage is at odds with the pronuclear axis, there weren't an awful lot of embryos, but there are almost no babies born in that. Only 11 babies out of the whole lot. This is a fair rarer case. It's only happening in about a third of the cases, which is different to what the data presented in the first paper is. And if you look at the case where there's alignment between the pronuclear axis and the cleavage axis, right? So cleavage, spindle, cells separate this way to give you two blastomeres. That's about 2/3 of all embryos are like that, and the majority of those are live births, suggesting that this arrangement is more common and more associated with live births than these two. And I think that this is an important one. This is a figure 5 of their paper, and they're basically in human fertilization, the centrosome, the cell's primary microtubule organizing center, is paternally inherited, meaning it's introduced into the egg by the sperm. And unlike the mouse model where the centrosomes are maternally inherited, human oocytes lack functional centromeres relying on the sperm to provide them for early development. And in this graphic, they show two things: a misalignment and effective age. So here, male pronucleus centrosomes, which lock the two pronuclei together and allow the cleavage plane, spindle axis this way, the fertilization plane here, so that you can see that the plane of the pronuclei and the cleavage plane are identical. And in this case, you have mononucleation, single nucleus in each blastomere. And the authors argue that with increasing maternal age or other forms of dysfunction, the centrosomes are misaligned inside the pronuclear development, and therefore the spindle is misaligned incorrectly. And so even though the cleavage furrow might be in the same way, that if there is misalignment, you get multinucleation in the two cell blastomeres. And you can see I'm struggling with this because it is quite a difficult thing to get your head around. And I just want to show a couple of quick papers at the end. First is the paper published 2022 showing the first mitotic division of human embryos is highly error prone. This is looking at the micronuclei, just as we've been talking about. But as they say, if you look at the incidence of live births between the mononucleated and the multinucleated, in fact, the live birth is quite similar. So even though you have multinucleation in some blastomeres suggesting some misalignment of the spindle phase with the cleavage furrow, it doesn't necessarily mean that it's detrimental to fertilization. And the second thing I just wanted to briefly talk about is the article published in 2019 by Nina Desari and Gill. They're just looking at the way the embryo divides the second cell into the fourth cell, the second cell, third, or the first division is 2 cells, the second division is to 3 cells, and the fourth division is into 4 cells. And this third division is important because I think that the cell that divides first from the 2 cell to 4 cell stage, these cells are earmarked to the epiblast, and the one that lags behind is destined to become defected. So there's 3 different combinations of 4 cell embryos, which have some impact of what's happening in the zygote in the 2 cell stage. And basically the embryos that are discarded here are very few, whereas more than half of these embryos are discarded. 80% of these embryos are frozen or transferred, as compared to only 50% for this. 87% blastocyst compared to 74, and expanded blastocyst 71 compared to 63. So there are some differences between them, but it's not an all or nothing thing. I also just need to recognize a great paper came out just recently, December 2025, by Coticchio, looking at the emerging role of the oocyte cortical domain, looking at the functions of microtubules in moving pronuclei around. I won't go into that for another paper, another talk I'd like to do, but I'd just like to recognize that the role of pronucleum and how they orientate inside the zygote is basically a function of the cortical domain and the microtubule system. And just one last, he also produced a great paper from time-lapse stuff for 2018 showing the breakdown, but in reality, this is the timing. See here, the polybody extrusion occurs very quickly, and then all the other events occur fairly quickly. The appearance of pronuclei, the alignment occurs very quickly, and then there's a long stage here when after there's a line before there is you start to have the breakdown. You see there's a distinct gap between the formation of the pronuclei and the alignment. This is where they're being aligned. This is where it's breaking down, and then there's very quickly after that breakdown, the cleavage. So whatever happens is in the formation of the pronuclei, and once they break down, cleavage occurs very quickly. And the last thing I just think is I found a great little paper published in 2022, also in the mouse, which might be the same as human. But if you take the polar body out of an embryo, so here they're arguing that the second polar body produces, here we report the second polar body in the mouse zygote may play a role in cell fate specification and post-implantation development. A subset of messenger RNAs encoding pluripotency related factors are enriched in the second polar body. These proteins may be synthesized in the polar body after fertilizations and transported to the zygote to form part of the animal whole. And the important point is, and I don't know whether this paper's been replicated or not, that I think maybe also for another video. But if you remove the second polar body from a mouse embryo, it will not form a baby. You'll form blastocyst, but you either get a trophectoderm or a runted embryo, but you don't get a live birth. And I think this paper really does need to be replicated. And also, no one can do it in the human, but I think maybe we should be looking at what happens to the second polar body and live birth. And just to finish up, I know it's a long talk, I'm actually ready for another cup of coffee. This is another video I'm preparing, but I thought I'd just throw this slide in because the outcome if you remove the second polar body and remove the ability to influence the animal vegetable cytoplasm is that you get a placenta or a runted fetus but no baby. It's very reminiscent of what happens with genomic imprinting. And there was a great paper produced in 1984. And I met Dr. Serrani, a very impressive man. But what they did in the mouse, they were able to take because the pronuclei are different, the male pronuclei in the mouse is larger than the female pronucleus. You can take the male pronucleus out and put a female pronucleus in. This is pretty impressive in 1984, being able to do this micromanipulation. But if you create an embryo with two female pronuclei, you get a genomic lethal form, which produces a small runted fetus and no placenta. And if you replace the female pronucleus with another male pronucleus, so it's an androgenic, they're also lethal, but what you get is all placenta and no fetus, almost like a hydatidiform mole. And the only way you can get a live birth in these mouse experiments is to have a female and male pronucleus. And so I'm leaving you with the impression that a lot of things are happening at the same time on the first cell stage. That is the influence of the polar body, which I think hasn't been really looked at in humans, and the relationship between how the embryo pronuclei align and first cleavage and cell fates. So just my last comments. Who would have thought that the orientation of two pronuclei relative to the polar body could impact the pregnancy rate? Most embryologists only consider checking the embryo on day one to ascertain the embryo is diploid, and they're not that worried about it. But staring in front of us was evidence to suggest whether the embryo is aligning itself correctly, or there is dysfunction inside the movement of the pronuclei. Most embryologists consider the time before gene activation to be an oocyte phase and of little value. And right in front of our eyes are critical clues as how the embryo is organizing itself. And I bet all the AI systems that are being used to generate embryo scores are looking at these arrangements in their assessment of their embryo score. Of late, the IVF industry has become obsessed with PGT-A and euploid assessment, and anything else is a minor value. And I think we're often throwing the baby out with the bathwater. And I tell you why, because if I look at this is a great paper. If you look at the ability of one blastomere to give rise to the epiblast, and how when this is dysfunction, you get a distorted embryo. The embryo is trying really hard to work out what to do with itself. So I say that there is currently some debate in how human embryos first cleaves. There is also new information suggesting that each blastomere may not be identical in their development pathways to the epiblast and fetal fates. The cytoplasm appears to produce differing proteins in relation to the sperm activation, and this process that governs the orientation of the first cleavage plane relative to the orientation of the pronuclear and polar body is staggering, in my view, of its implications. IVF in this data suggests to behave in a similar fashion, although I think there are more papers in the future will actually clarify whether this is correct or not. and maternal age appears to have an impact on pronuclear development and how the embryo cleaves in the first stage, and the implications of euploidy, aneuploidy, and multinucleation. When I look at these publications, I sense that there are some profound basic events happening during pronuclear development that specifically impact on blastocyst development and implantation. These events are not limiting, since ongoing pregnancies occur in alternative scenarios. There's a lot to be said recently about the ability of the embryo to self-correct, say with mosaicism, but I think also too in desperately trying to find cells to form the epiblast. However, as embryologists, we should respect that all things do not happen by chance, and that each sequence is important. And we should look closely at what we can observe during pronuclear development and view blastocyst morphology and dynamics in terms of their etiology, and just not ignore what the embryo may be trying to tell you. My final word is that none of the above reports and observations are definitively critical. There appears to be considerable flexibility in the developmental pathways. What we need to be aware of are the multiple steps that allow the pathways of individual cells to take in their destination to fetus and placenta. It's kind of interesting. I've been doing embryology for more than 50 years. When I look at these papers and think about them over a glass of wine, etc., you can see that each cell is desperately trying to find its place in the embryo and desperately trying to find its way into either the outer cell, the trophectoderm, but desperately trying to get into the epiblast. And it's almost as if all these individual cells that they're dividing are desperately trying to find a way to form the fetus. And there's something really quite profound, almost magical, about the desire for each individual cell with its own legacy, to keep trying to find a pathway to develop into the epiblast. So I'll just come back to what I stated early on. This article is a summary of many of the key zygotic events that occur out of sight. When all the key axes are in alignments, like the stars, like the polar body axis, the pronuclear axis, and then the cleavage plane is in alignment with these, then the embryo would more likely to be euploid and more likely to have the preferred cytoplasmic properties, which allow the embryo to go down its preferred pathway with minimal interventions and minimal sorting and correction, and therefore they're more likely to lead to pregnancy and a live birth. I think this has been a great paper. I spent about 2 weeks trying to prepare it, and I've learnt more in this paper than I have on this video than I have in most other things. So just to finish up, my name's James Danger. I'm an embryologist with 50 years experience. These videos are my attempt to bridge the gap between information you may need for your fertility treatment and the most recent publications available to your staff. I am not a medical person. This information is not medical advice. But what questions to ask about not medical advice, but may help you in knowing what questions to ask about your treatment with your doctor, and maybe help in understanding the advice you receive. If you like what I'm doing, press subscribe, press like, tell everybody else. But I hope basically that it helps you on your journey. Just a quick plug for my other work, my sponsor, which I'm sponsoring myself, my Fertility QA program. And just my last slide, as always, just to thank you for your attention if you got this far. Now, it's a long journey for this video, and I'm very impressed you got to this point. But I do recognize IVF as a personal journey that involves the action of many ideas, function, techniques, and people, and it helps to understand all aspects of that journey. I hope this information may be of value. And if you like what I'm doing, please tell all your mates and friends. And at this point in time, I'd like to stop and say goodbye.